Hey everyone, my name is Daniel Gataulin. I'm one of the senior research analysts covering biotech here at Chardan. It is my pleasure to introduce Jay Ducker, the CEO of EyePoint Therapeutics, to our virtual ophthalmology summit, where the focus of today's discussion is going to be on AMD and diabetes-related diseases. Jay, welcome.
Thank you, Daniel. Happy to be here.
Excellent. So to get things going, let's spend a few minutes and if you could introduce yourself and introduce EyePoint along with the recent milestones.
Sure. Yeah, I'm Jay Duker. I'm the CEO and president of EyePoint. I've been in this role for a little over a year. Prior to that, I was chief operating officer. And in my former life, I was chair of the Department of Ophthalmology at Tufts University in Boston for over 20 years. I am still a practicing retina specialist, and I think that gives me quite a lot of insight into the way retina specialists and the retina community and the payers would view the ability for a company to provide sustained release in these VEGF-mediated diseases. So that's what EyePoint is focused on right now. We are a drug delivery company to the back of the eye, and our lead product is called DURAVYU. It's currently in 3 phase 2 trials in VEGF-mediated diseases. Those are wet AMD, non-proliferative diabetic retinopathy, and diabetic macular edema. DURAVYU, which I think we'll talk a little bit more about as the session goes on, but briefly, it is a small molecule tyrosine kinase inhibitor called vorolanib, which has excellent activity against all the VEGF receptors, and it's in our proprietary delivery system, which we call Durasert E. And we're very excited about the phase 2 results and the fact that we anticipate starting 2 pivotal trials in the very near future in wet age-related macular degeneration.
Got it. Excellent. And DURAVYU is also called EYP-1901, so forgive me if I by habit refer to it as 1901. So in any case,
I still do occasionally also, so no problem.
Yeah. So you mentioned the Durasert technology. Let's talk about that first. Can you describe what it is, what its capabilities are? Why do you believe it's de-risked? And I guess specifically to that application, how do you control the dose with Durasert?
So Durasert has actually been around for a while. And one of the reasons we think it's really highly de-risked is it's already in 4 FDA-approved products. The former iterations of Durasert were not completely bioerodible. They consist of a core matrix of a drug with excipients to pull the drug together, and they were wrapped in a polyamide shell. And the polyamide is impermeable to the drug and is also non-erodible. So the polyamide shells for the previous iterations of the products would last essentially forever, although they're quite inert. There are pores at one or both ends of the cylinders of the prior iterations, and those pores allowed for simple diffusion of the drug into the vitreous cavity, then across the retina. These inserts feature what's called zero-order kinetic release, which means once a steady state has been achieved, they're releasing against a gradient. And as that gradient is steady state, there's then a steady state release of the drug. That steady state avoids the typical Cmax, Cmin that you would see from other types of drug delivery and enables what's been referred to as microdosing. For example, if you look at our YUTIQ product, it contains only 0.18 milligrams of corticosteroid, but it lasts 3 years in the human eye, and it can control uveitis patients who have inflammation. 60% of them can be controlled with a single YUTIQ injection for that 3 years. So a relatively very low dose of corticosteroid can be effective. And we think that will be also true of DURAVYU, formerly known as EYP-1901, with the VEGF-mediated diseases as well.
Got it. Okay. And I guess the second component of DURAVYU is the TKI, tyrosine kinase inhibitor, vorolanib. Before we dive into that, can you talk about the mechanism of tyrosine kinase inhibition and more specifically its role in wet AMD, and what is the interplay between that and how it complements anti-VEGFs?
So the VEGF-mediated diseases obviously all feature high levels of VEGF in the eye, and the prior treatments have been directed toward blocking the ligands, and they do it quite effectively. What the tyrosine kinase inhibitors do is block at the receptor level intracellularly. So they're generally small molecules that can get into the cell and block the receptors. So the advantages, again, small molecules have some advantage. They're also then, in our case, able to be put in a delivery system, Durasert E, that allows them to last many months in the eye. But the other advantage of blocking at the receptor level is that you can block all isoforms of VEGF, not just VEGF A or B, but all isoforms, which theoretically can be advantageous. TKIs generally have other effects also, depending on the dose that you use. Some of them would be referred to as off-target effects, and the good news is there doesn't seem to be any reported ocular side effects from the use of any of the TKIs for VEGF-mediated diseases. But some of the off-target effects can be beneficial. For example, vorolanib blocks PDGF, which means theoretically we should be able to reduce the fibrosis that occurs in some of these diseases. And in the long term, fibrosis is one of the reasons that wet AMD patients lose vision, even when they're under good control with anti-VEGF agents. One other feature of vorolanib of note is that at the levels we're using, it does not block TIE2. You don't want to block TIE2. TIE2 agonism is now used in the FDA-approved product faricimab or Vabysmo in the sense that it blocks angiopoietin-2. If you block TIE2, you're going to upregulate angiopoietin-2, which causes instability of the retinal vasculature. And unlike some of the other TKIs that are currently under study, vorolanib doesn't affect TIE2.
Got it. Okay. And in terms of vorolanib and its validation in wet AMD, I understand there have been several studies, including some earlier studies with oral dosing. Can you give us a brief summary of the data from those?
So vorolanib was originally developed by the same protein chemists that developed sunitinib, or Sutent, which is FDA approved as an oral agent for kidney cancer. And the thought was that vorolanib would have fewer off-target systemic side effects than Sutent. And the developers had the idea that they might try using it initially in a large market like wet AMD. And in fact, that's what they did. There were two phase 1 and a phase 2 trial, two trials run. And at the time, the molecule was called X82, looking at the use orally of vorolanib in wet age-related macular degeneration. And it did show really, quite frankly, good efficacy. But the problem was that there was systemic side effects, mostly hepatobiliary side effects, that prevented the phase 2 trial from ever really advancing. And so that was one of the advantages we saw in vorolanib, is that the FDA had already seen it. It already had an IND. It wasn't approved in the United States, but that data enabled us to, speed our program in advancing it and give us good background and evidence that in an insert form, vorolanib would be effective. The other advantage to vorolanib over, for example, axitinib, which is being used by other companies for VEGF-mediated diseases, is vorolanib has excellent IP protection. There is composition matter protection out to 2037, which can probably be extended to 2042. And since it's never been FDA approved in the United States, if we were to get the first approval in wet AMD, that would also give us that new drug exemption that comes with the first approval. So we have quite good IP protection around it. And of course, the molecule itself, vorolanib, was able to be fit into the Durasert E delivery system, the erodible form of Durasert, quite nicely. So it really checked a lot of boxes for us, and it's turned out to be quite an advantageous TKI.
Mm-hmm. Okay, got it. So you mentioned a couple of differentiators of vorolanib versus other TKIs, specifically axitinib. I think you mentioned the TIE2 differentiation is one, the IP. Anything else? And maybe in terms of the inhibition component itself, how does that compare?
Well, so one advantage axitinib has theoretically over vorolanib is it does have more activity from an IC50 perspective for the VEGF receptor 2. And what that would correspond to, of course, would be in clinical practice, one could use a lower dose to get the same effect. And in fact, I do believe that's what you're seeing in the clinical trials. For example, Ocular Therapeutics is using a dose of 450 micrograms of axitinib in their phase 3 trial. We're planning on going forward with a dose of 2.7 milligrams. Beyond that, I think all the advantages really go to vorolanib. We talked about IP, we talked about Tie2. The other advantage has to do with the way sustained release products are developed. And you need the right kind of solubility. If a drug is too insoluble, then it will have a problem diffusing into the eye. And axitinib is more insoluble than vorolanib. Now, one of the things that we designed with DuraView was we designed the matrix to hold the vorolanib for the length of time that we want, which is a minimum of 6 months in the human eye. After that, the matrix, which is bioerodible, slowly goes away over the next several months. Because axitinib is less soluble, that if your matrix goes away in, say, 6 months, you may have free drug in the eye. And obviously, once you have free drug in the eye, it may migrate and it may not release at a rate that is effective, or may even release at a rate that causes toxicity because you're no longer controlling that rate of release. So again, the theoretical advantage of having a better activity against receptors can be overcome by dosing. And the truth is, once a receptor is blocked, it's blocked. And we looked at this in a paper that's actually not published. Well, we did an experiment using what's called the CAM model. It's the choriontilic chick model. It's a model of neovascularization. And we took the 3 TKIs that have been used in the back of the eye, axitinib, vorolanib, and sunitinib. And we did it against Avastin in the CAM model. And we used just the IC50, not even a multiple of the IC50. And all 3 TKIs actually blocked neovascularization better than Avastin did. Now, once we increased it to 200 times IC50, we got the exact same result, suggesting once you've got that receptor blocked, excess is not going to give you a better clinical result, but in fact may give you increased off-target effects, which you got to be careful of.
I see, makes sense. Okay, and in view of neurobiologics that are being developed as well for longer treatment intervals, what's your view on those, and where do you see the value add for DuraView?
Sure. So we already talked a little bit about Vabysmo and the advantage that it does block angiopoietin 2. And it does appear to have some increased longevity and drying effect in some patients versus the standard 2 milligram Eylea dose. And some of this real world data is now coming out. And more recently, we saw a presentation that was real world data from RCA, which is one of the largest retina groups in the United States, if not the largest. And they showed in treat and extend protocol, they were getting about 8 days longer with Vabysmo than they were with 2 milligram Eylea. So 8 days, you might argue, not a long time, but Vabysmo is $1 billion product now. And what it's telling us is the retina community is craving longer acting agents, and we'll use them even for 8 days. Now, we also have high dose Eylea, and we don't have quite as much experience with high dose Eylea yet as we do with Vabysmo. But the suspicion is based on the pharmacokinetics that there will be a little bit more longevity in that treat and extend protocol using HD Eylea. But none of these products can reliably go 6 months or longer like we believe DuraView will. And so that's the real advantage that we have is we've got consistent drug release for we think a minimum of 6 months in humans. And that will enable a flexible dosing regimen for many doctors and patients should the drug be approved, safe, effective, tolerable, and with an every 6 months regimen, which is what we're looking at in the pivotal trials. So I think any innovation in the retina space is great for patients, and we do have 2 new agents, but I think they're incremental improvement. And I would summarize by saying, first, the number of eyes with these agents that are going to be able to get out 4, 5, 6 months reliably is relatively small. And the upside is still true, is that there's still a lot of patients with wet age-related macular degeneration that, despite Vabysmo and high-dose Eylea, still need to be treated every 4, 5, 6, 7 weeks to maintain their vision. So there's still that group of patients that we refer to as frequent flyers that are still out there despite these new drugs.
I see. Got it. Okay, so you're in that unique position as a CEO of a company advancing novel therapeutic for wet AMD, and you've been on the other side as a treating retinal specialist. So you see kind of both sides to the story. So from your experience, what kind of feedback are you hearing from patients and maybe your colleagues in the space? With their real life experience with those biologics that we just discussed, and maybe from your personal view, not just the other studies, but your personal experience in terms of patients asking for something that is more durable.
Yeah, so that is kind of the holy grail right now, is the more durability without sacrificing much vision and without sacrificing safety. And I'd like to talk about safety first, because that's really important to retina specialists, especially in the United States, where there are certain drugs that have been associated with intraocular inflammation that have made some retina specialists reluctant to use them. So the good news about the review is the safety record's really, really good. We've had over 190 patients receive the drug with no ocular or systemic SAEs and no sign of any posterior segment inflammation or migration of the insert or anything like that. So far, that safety box has really been checked. And of course, we have about 150 patients who were treated with X82 vorolanib orally that had no ocular problems either. So safety is really critical, because if you don't have it, it's going to be really hard to introduce a new drug into the market. The longevity issue is that the retina specialists are just overwhelmed with injections. And while we like the response of the agents that we have, they just don't last long enough. And of course, if you're a patient who doesn't live near a big city, you may be driving 2 hours or 3 hours each way monthly to get an injection. That's really, really hard to do. And we think some of the problems that patients have maintaining their vision in wet AMD is not because the drugs necessarily aren't working. It's that they just can't get in frequently enough, monthly or 6 weeks, to get the required dosing. And that's why I think doctors and patients are very excited about the opportunity to go longer and safely. And again, if we talk a little bit now about the phase 2 data, the phase 2 wet AMD trial showed that 6 months after DURAVYU went in, at 8 months in the trial, there was really essentially no statistical difference between the visual outcomes in the DURAVYU patients and the visual outcomes in the patients treated with 2 milligram on-label Eylea. So that durability and visual acuity box, we think in the phase 2 has largely been checked, and we hope to do at least as well, if not even better, in the pivotal trials.
Got it. Okay. Yeah. So yeah, let's shift gears and talk about the phase 2 trial. So you reported the results, very impressive results. Before we dive into all the details, can you summarize key aspects of the trial design?
Sure. So the phase 2 was obviously multi-center randomized mass trial. It enrolled 160 patients, all of whom had previously treated wet age-related macular degeneration. They were then randomized to approximately 2 milligram dose of DURAVYU versus a 3 milligram dose of DURAVYU versus on-label Eylea. And despite the fact that they were previously treated, everybody in the study got reloaded with Eylea. So on day 1, they got an Eylea shot. At week 4, they got an Eylea shot. At week 8, they got an Eylea shot. And then 30 minutes later, they either got their DURAVYU, 2 milligrams or 3 milligrams, or they got a sham injection for masking. Then the DURAVYU arms were not re-dosed with DURAVYU during the trial. The patients were then seen monthly. And one interesting aspect of the trial is there was supplemental criteria, meaning if a patient had disease activity, they should be supplemented with an Eylea injection. But the doctor that was determining that was masked. And therefore, there were actually supplements given in the Eylea arm. despite the fact that those patients were getting every other month Eylea. And one of the things that illustrates was that we ended up enrolling a pretty tough to treat population in DAVIO 2, the phase 2 trial. On average, the patients had about 10 injections in the year leading into the trial, which is a lot. Most patients in the real world have about, on average, 6 injections. So it was a tough to treat group, and that I think makes the results even more impressive. So we did report the 8-month results, which is 6 months after DURAVYU went in, and we reported that last December. And just recently, we revealed some of the 12-month data, and that was at an R&D day that we did several months ago. And the 12-month data showed continued safety, which is excellent, and also showed that from a visual acuity perspective, the patients were maintaining the vision. And out a year, at that point, we would have expected that basically all the inserts would have been depleted for vorolanib. So we would expect at the end to see a slight bump in the number of rescue injections given and maybe a slight drop in visual acuity. But so far, the data really shows complete stability with the higher dose, the 3 milligram dose, having the exact same vision at that point as did the arm.
Okay, and so in addition to vision preservation, being able to control the fluid is also considered to be very important in wet AMD treatment. Can you comment on what you saw with CST effects?
Sure. So again, the fluid is measured by OCT. The CST means central subfield thickness, and that's a data point that the machine gives you non-invasively. And normal is about up to 280 microns. The standard deviation of the test is about 10 microns. And we do know there's some fluctuation in fluid, and in a little bit of subretinal fluid in a wet AMD patient is not necessarily a bad thing. And there's some patients, even with monthly injections of Vabysmo or high-dose Eylea, you can't get rid of all the fluid. You just can't. And that's, accepted that there's that can happen. So when you talk to clinicians about fluid, we use fluid as a biomarker for VEGF activity. So if you have an eye that's been running 300 microns with monthly injections, and all of a sudden they're 350 microns, you'd say, well, they're not in control anymore. And that is, again, one of the big points we make at each visit to determine what's the right treatment interval, the right drug that we're giving. The FDA, interestingly, it's not that they don't care about OCT, but they don't care as much as the clinicians do. But the good news is in the trial is that the anatomic control using DURAVYU was excellent, and that the difference at month 8 between the DURAVYU arms and the Eylea on label was 10 to 15 microns, just a small amount that's hardly noticeable just looking at an OCT. And we had talked to physicians prior to the result and, asked them, what anatomic control would you like to see with more durability? What fluid would you allow? And, we heard anywhere from 25 to 50 microns was going to be okay. So we bested that quite well. The other thing that was interesting, if you look at the OCT data from DAVIO 2, is after month 4, the fluid levels were basically stable. There was no sawtooth pattern, and there was no increase at the end, meaning if there was an increase in fluid at the end, you would hypothesize that the inserts were losing control. And that wasn't the case. It really remained stable. Even, we showed the 12-month data as well, up to, month 12, there was really quite a lot of stability in the anatomy. And so what we can tell the retina community is you can get more longevity. And we think the majority of eyes, it looked like at least 2/3 out to month 8 without any supplement. But you don't have to sacrifice vision, and you don't have to sacrifice anatomy.
Got it. Okay. So to kind of bring it back together, for the primary endpoint, which I believe was blended 28-32 week,
Right.
What effect did you see on BCVA and reduction in treatment burden?
So the endpoint of non-inferiority change in visual acuity is the endpoint that the FDA looks for in the pivotal trials. The blended endpoint is something that the FDA asks for, and so we did it in the phase 2, and we will be doing a blended visual acuity endpoint in the pivotal trials as well. So at the 8-month visit, the higher dose, 3 milligrams, was 0.4 letters worse than Eylea. And remember, on the eye chart, there's 5 letters to a line. and the
Mhm.
2 milligram was 0.3 letters worse than Eylea. But the statistics on it for a difference, they were the same to Eylea to a p-value of 0.0009 or less. So they were statistically identical. Reduction in treatment burden, there's 2 ways to measure it. You can measure it retrospectively. What did the patients get leading into the trial? Or you could do it prospectively, which is compared to the Eylea control arm, which was
Mhm.
mandated to get 3 injections over 6 months after the DURAVYU went in, about 80% reduction any way you measure it. And so significant reduction in treatment burden. And we took some eyes that were getting treated monthly and maintained DURAVYU alone without any supplements. Now, there were eyes that did get supplemented, but if you look at the swim lane charts, you can go back and look at the rate that they were getting treated leading into the trial. And even with the supplements that they received in general, it was at a much lower rate of injection and therefore visit burden for those patients, that if these data hold and we get approved, how it will be used in the real world.
Got it. Okay. So I believe next month you will be presenting full 12 months data.
Yes, we have.
I'm sorry.
Oh, I said yes. We have several presentations on the 12-month data coming up.
Yes. So was curious what additional analysis should we look out for?
Well, I think we really talked about the top line already, which is visual acuity, and the fact that the inserts were not re-injected, yet about half the patients were supplement free out to that 12-month visit. We've already talked about that, which is pretty impressive. Safety, again, we mentioned no new safety issues. We will show some subgroup analysis, and one of the other analyses we look forward to showing is that, remember I said that the doctor that determined whether a supplemental injection was necessary was masked. So they check box at every visit, meets criteria, didn't meet criteria. So we're going to show the number of eyes in the Eylea arm that met the criteria for supplement compared to the DURAVYU arms. And I think that the data will show, quite surprisingly, there's not a big difference, suggesting that, again, our drug you can think about it, is replace, every other month Eylea in a lot of patients. So that's something I think of interest.
Okay, got it. And let's talk about your new phase, the upcoming phase 3. Can you talk about the key aspects of the phase 3 design and maybe some of the key learnings from DAVIO 2 that you're implementing?
So we're about to start 2 pivotal trials, the Lugano trial and Lucia trial. They're very similar, if not identical, trials at this point. And they were based around multiple interactions that we had with the FDA starting in 2022, when we had a Type C meeting and culminating in the end of phase 2 meeting that we had several months ago, along with some other communications that we've had with the FDA. And so these trials were designed really to meet the FDA requirements for non-inferiority trials. And so they will consist, each trial will have approximately 400 patients. There will be 1 dose of DURAVYU, 2.7 milligrams against Eylea control. The randomization will be 1-to-1. And the major differences in the pivotal trials from the phase 2 DAVIO 2 trial are, first of all, the primary endpoint will be at approximately 1 year. It's going to be a mixed 52, 56-week visual acuity endpoint, and that's mandated by the FDA because we're doing a load in everybody again. Everybody's going to get reloaded. Number 2 difference, we're going to use the 2.7 milligram dose. We've actually tested that dose in the DME trial, but that is a higher payload insert that we have developed, and that's going to be the going forward dose. We're going to re-inject every 6 months DURAVYU. And we're trying to work for a label of every 6-month dosing. That is a difference. The other difference is the patient population. We're going to be enrolling also naive patients along with previously treated. And that we believe we expect will not only speed enrollment and give us a broader label and more acceptance across the world, but will also, we believe, increase the results. Because in DAVIO 2, the previously treated patients that we enrolled were heavily pretreated. They needed a lot of injections, yet they were a group of patients that needed continuous treatment. You can tell by the OCTs. We got very few patients on the other side of the equation that don't need a lot of treatment. And we think our drug will do very well with them. So by enrolling naive patients, we also will get some of those patients who need, let's call it, less intensive anti-VEGF therapy, and we expect to do very well with them. So we are on target to dose and obviously randomize the first patient in the second half of this year in the first trial, which is the Lugano trial. The Lucia trial should start several months afterwards.
Okay, got it, understand. And just to clarify, at 6 months, the patients will receive the second dose of Duravyu, correct? They will not be reloaded with Eylea.
No, there's no need to reload them. Because obviously the supplemental criteria will be there. We'll have supplements. If an eye has disease activity, it will get supplemented. And so if an eye is doing well, there's no need to give them a supplemental injection.
Got it. Okay. But at the same time, the eyes that would have been supplemented by 6 months, they would still get the second dose of Duravyu.
Yeah, everybody will get that second dose of Duravyu essentially at month 8. 6 months after the first dose goes in. And again, they will be 2-year trials because we want to get a label for redosing fully, and we want to show the 2-year safety to get a complete label. But the FDA has agreed that we can submit the NDA after 1 year of data.
Got it. And with respect to real life scenario at steady state, so to speak, would you say the phase 3 design represents what the patient treatment paradigm would look like?
Well, it's hard to say that because, as you know, and anybody who knows this field is retina specialists, because we have the ability to monitor the disease in the patient right in front of us, we tend to modify the disease protocols in individualized therapy. The good news about our study design is it's against the 2 milligram Eylea on label, which everybody knows how that performs. And this is the same study design that essentially the last 5 anti-VEGFs have been approved with. So the retina community is going to understand the results and how to use them quite well. Now, whether a doctor chooses to do every 6 months or individualized therapy, because I just said we had some patients that could go a year, and they may do that. And that will be up to the individual physician and the doctor, and whether they're comfortable letting a patient go 6 or 8 or 9 months between visits. Again, I suspect at the beginning, the doctors will watch the patients more carefully, but they may eventually, especially with the use of home OCT, be comfortable letting some of the patients go quite long between visits, provided they're comfortable that the control will be there. The other important point, and we've talked about supplements, is that a supplemental injection for us is not a failure. No drug works perfectly, and that reduction in treatment and visit burden without sacrificing vision and safety is what we're going for. So if I have a patient that I have to treat monthly and I use Duravyu, and that patient requires 1 or 2 supplements over the next year, that's not a problem. I've reduced the treatment burden significantly, and both patient, doctor, and probably payers are going to be happy about that. So that a supplemental injection per se does not represent a failure of our drug, because the goal here is to allow doctors and patients the flexibility to go longer between visits without sacrificing vision and safety.
Got it. Okay. And one last question on wet AMD. With respect to Duravyu manufacturing, can you comment on your capabilities to supply the drug both for the phase 3 program as well as the potential commercial supply?
So the phase 3 program, we're really all set with that. We manufacture the drug here in our office in Watertown, and we have clean rooms to do that. And, we've been manufacturing commercial drug here for many years, in particular YUTIQ. But we're actually building a manufacturing facility. In fact, it's completed in a town about 45 minutes west of here called Northbridge, Massachusetts. That's state-of-the-art, beautiful, large clean rooms that will be able to accommodate the commercial needs. And we'll be able to make well over 1 million inserts a year in Northbridge when it's actually up and running. So the phase 3 trials will be supplied and have been supplied by manufacturer down here in Watertown, but commercial supply in potentially other indications, for example, DME or eventually RVO, in the future, if we do pivotal trials in those indications, would probably be supplied out of the Northbridge facility.
Okay, got it. I wanted to spend just a few minutes on your other programs, NPDR and DME. So you reported data for PAVIA trial for NPDR that unfortunately did not perform to some of the expectations. Can you speculate why do you think that is? And maybe is there any aspect of a trial design that you could have changed?
So yeah, it certainly was disappointing. On the other hand, our wet AMD data really stands on its own, and the wet AMD data is excellent. And that's really where, the big market is. As you know, the NPDR market, it has to be carved out. It's not there yet. There are 2 FDA-approved drugs, but hardly anybody uses them. We think the most likely explanation as to why the results were not what we expected in NPDR is off target. I talked about this before, that the TKIs do have effects on other receptors, and there appear to be several potential receptors that vorolanib and other TKIs may block, which will then produce vascular instability in diabetic retinopathy, but are not a factor in wet AMD. We're doing a little more work on that, but we think that essentially the benefit of blocking the VEGF receptors with vorolanib was counterbalanced by the likely off target of 1 or more blockages of other receptors that early in NPDR you don't want to block. So we're not going forward with the NPDR program at this point. I think it's certainly possible that in the future, a partner may look at the data and want to revive it. But it doesn't make sense for us to spend our resources and time on it right now. DME, we've got the Verona trial that's fully enrolled and on target to present top-line data in the first quarter of next year. And remember, DME is an exudative disease like wet AMD. There's active fluid being leaked from abnormal blood vessels, unlike NPDR, where there are markers in the fundus, in the retina for the disease, but no leakage. And so that we're optimistic that we should see that same type of maintenance benefit that we've seen in wet AMD in DME. As for changes in trial design, we talked about, would it have made a difference in NPDR if we had loaded the patients? The answer is yes. I think if we had loaded the patients, we would have gotten most likely a positive result, but we wouldn't have a commercially viable drug. This isn't just about getting a positive result. This is about drug development in helping people in a fashion that makes sense. So if we had loaded patients, that would have been 3 Eylea injections plus every 6 months DuraView. In the first year, and 5 injections. And the main reason that Lucentis and Eylea aren't being used in this disease is too many injections. So we might have gotten a different result, but I don't believe we would have a commercially viable drug.
Understand. Okay, makes sense. All right, and I guess in the last minute or so, can you give us a summary of where you are in terms of your cash and runway?
Yeah, so we have cash runway into mid-2026, which is what appears we'll have top-line data from the pivotal trials at the same time. Last was about, we reported about $280 million in cash and cash equivalents. So balance sheet's looking very good. And obviously, we've had no debt. We retired the debt when we sold YUTIQ to Alimera over a year ago.
Okay, got it. And maybe in a sentence or two to conclude, what do you think investors are still missing about EyePoint story?
We've got a drug that is being looked at in a multi-billion-dollar market that is about to start 2 phase 3 trials that are funded, and we had excellent phase 2 data, both efficacy and safety. I think that, in a nutshell, suggests that investors are not viewing our value the right way.
Got it. Excellent. All right. Well, thank you very much, Jade. It's been a pleasure talking to you, and I look forward to connecting again in the future. Thank you.
Bye.